Yishan to do his defense today. And so probably it's too early, so not many students are here. So Yishan joined the Ph.D. program about 5 years ago, fresh from undergraduate. So he was the top students from Wuhan University in statistics, and then with minor in computer science. So during the last 5 years, almost 5 years, and so he did extremely well. So he has published extensively over the last several years, and in particular, he has 2 Nature Communication publications in metagenomic and 2 recap papers. And so the recap, we all know, is one of the top conference in the field with less than 20% acceptance rate. So, according to our kind of standard, it's 2 papers, it's decent papers, and so I think he now completed 3 PhDs in the last 5 years. So anyways, it's great to have him, and he contributed a lot to the research.
Okay, so thanks Professor for the introduction, and thanks all of you being here. Today my defense title is Constructing the Metagenome-Assembled Genomes and Mobile Genetic Element Host Interactions using Metagenomic IC. So when we look inside the human body, we will find a lot of small living things called microbes. In fact, the microbes spread out across different parts of our bodies, like our mouth, our skin, and our gut. There are very diverse microorganisms, including the prokaryotes, eukaryotes, viruses, etc., all living with us. Most of the microbes reside in gastrointestinal tract, and remarkably, there are billions of microbes in just a gram of real human feces. With the microbiota and human cells in almost equal numbers, we are really linked together in a perfect balance with microbes. Most importantly, the microbiome in human body has a significant impact on our health and is closely associated with multiple diseases. For example, there are lots of studies showing that when the microbes in human gut are not in balance, which we call the gut dysbiosis, it can lead to severe gastrointestinal diseases or even malignancies, such as the inflammatory bowel disease and colorectal cancer. Actually, millions of patients in the United States are suffering from these diseases. The gut dysbiosis can also cause metabolic disorders, including the obesity and type 2 diabetes. Another very well-known concept regarding the gut microbes is so-called gut-brain axis. The gut-brain axis is not a new concept and can be traced back to early 20th century when the scientists have found that the toxic content in the gut can have a profound impact on our brain health. Nowadays, the gut-brain axis is getting more and more attention since the recent studies show that this special mechanism may play a very important role in cognitive disorders, such as the Parkinson, the Alzheimer, and autism. Because of the importance of the microbiome, many initiatives and resources have started to support the microbiome investigation, including the famous Human Microbiome Project and the recent Human Biome Project starting last year. Moving from the human-centric studies, the Earth and the Ocean Microbiome Project further expand our horizon to diverse environments with great impact on our climates and our ecosystems. As we deeply investigate the microbial communities, we can find several questions that challenge our current understanding. At the root of the quest is the most fundamental question: who is in there? This question calls us to characterize the complicated structures of the microbial communities, including the identification of larval viruses and other microbes. Moving further, we must deal with what are they doing. This drives us to identify the micro interactions, such as the virus host interactions within a specific ecosystem, such as the human gut. It's not just about who they are, but also about their metabolic pathways and how they communicate. And finally, how are they doing? This question seeks to find the presence of microbes and viral host interactions correlate with disease states. And this can help us develop the new treatments as well as the preventive strategies. So these three questions remain challenging. Even for the most fundamental question, who is there? This is not a naive question, since most of the microorganisms cannot be cultured in lab, and culturing can never capture the full spectrum of microbial diversity. This problem remained unsolved for a long time, until the metagenomics has completely changed our understanding of the microbial world. Metagenomics does not rely on traditional culturing-based methods to explore the microbiome. Instead, it employs the genomic sequencing to sequence DNA from all the organisms in a given environmental sample, all at once. This can give us a comprehensive overview of the microbial community's diversity without isolating and culturing individual species. In the field of metagenomics, the metagenomic high-seq or meta-high-seq experiment stands out as a cutting-edge technique. In the high-seq experiment, we take two samples from the same microbial community, like the human gut, and these two samples will be sequenced using two different sequencing technologies. We use the shotgun sequencing to capture DNAs from all organisms present in the sample, and use the high-seq sequencing to map the physical interactions between DNA segments. Specifically, in the first sample, we break down the cells, fragment genomes of all the microbes into smaller pieces, and sequence all the DNA present in the sample into the shotgun reads. These shotgun reads represent a mix of all genomic materials, including that from prokaryotes, eukaryotes, viruses, and plasmids. After sequencing, we assemble these shotgun reads into longer and contiguous sequences, known as contigs. But there's a catch here. These assembled contigs are still incomplete and cannot represent the entire genomes. So we group these assembled contigs into clusters, which we call the draft genomic beans, and we hope each bean can represent the full genome of one species. This clustering process is called contig-inning. In fact, the contig-inning process is exactly how we begin to answer the question, who is in there, by directly retrieving the complete genomes from our microbial community. But how do we know which contig belongs to which genome? That's where the high-seq sequencing comes in. The high-seq sequencing can tell us whether two contigs come from the same genome and help us construct reliable contig-to-contig relationships. High-seq sequencing is originally designed to explore the three-dimensional folding of the genomes. Here is how it works. For the second sample, we keep cells intact. This is a very important point. Inside each intact cell, we freeze the DNA sequences that are close together. This is done by cross-linking the DNA, cutting it into fragments using restriction enzymes, and then joining adjacent sequences to create chimerical junctions. This process is called proximity ligation. Each chimerical junction is two DNA fragments from different genomic loci joined together, indicating they are close in the same cell. Then, after we create the chimerical junctions, we open the cells up and use the parallel sequencing to read both ends of each junction, generating two linked reads per junction. Since these two DNA fragments are labels inside the cell, the paired reads also come from the same cell, right? So based on this property, we can map back the paired and high-seq reads to contigs assembled from the shotgun sequencing. If we find a pair of high-seq reads mapped to two different contigs, it suggests that the two contigs may come from the same cell, or in other words, come from the same genome, right? we count how often this happens. These are our raw Hi-C contacts between contigs. For example, the raw Hi-C contacts between contig 1 and contig 2 are 2, right? In this way, we can generate the raw Hi-C contact maps in a graph format, where the nodes are our assembled contigs and edges are weighted by the raw Hi-C contacts. The raw Hi-C contact maps can also be displayed as a weighted adjacency matrix, which we call the raw Hi-C contact matrix. In this matrix, the row and column represent contigs, and each entry shows the raw Hi-C contacts between two contigs. Based on the Hi-C contact maps, you can do the contig binning, which is exactly a graph clustering problem, right? The contig binning step helps us generate draft genomic bins, moving us to identify the diverse microbial species in our sample and finding out who is in there. But the capability of meta-Hi-C experiments do not end here. Let's say if a phage has infected a bacterium. The DNA of both phage and host are in the same cell and can be cross-linked, generating chimeric junctions, which can be sequenced into the paired end Hi series. Therefore, if we see the Hi-C contacts linking viral contigs and prokaryotic or eukaryotic contigs, it suggests infections may happen. This insight allows us to answer the question, what are they doing? By revealing the interactions between the mobile genetic elements and their hosts, right? Although the meta-Hi-C method is promising, we face three major computational challenges in meta-Hi-C data analysis. First, normalization. Like most of the biological experiments, meta-Hi-C is not immune to experimental voices. So normalization is very important to eliminate these systematic biases for better downstream analysis. Next, we want to effectively cluster contigs into draft genomic bins, and we hope each bin can represent the complete genome of one species with as no contamination as possible. Our third challenge is how to accurately predict the virus host interactions. It's essential to distinguish the true interactions from false positive linkages within the virus host Hi-C linkages. In the following section, I will introduce my solutions to these three challenges. So let's start from normalization. Our motivation for implementing the normalization comes from the challenges encountered in binning contigs using raw Hi-C contact maps. This figure shows the clustering performance on a synthetic meta-Hi-C dataset, which is derived from a manually curated microbial community through culturing a mix of 13 yeast species. Since the reference genomes of all species present in the community are known, we can know the species identity of all assembled contigs by aligning the contigs to reference genomes, right? These contigs labels can be used to evaluate the clustering performance. The blue bar shows us the results of a method called VNB, a binning approach that does not rely on the Hi-C information, while the brown bar uses the Hi-C context combined with the latent graph clustering, which is a very popular graph clustering algorithm. Unluckily, we find that the raw Hi-C context or raw Hi-C contact maps cannot provide us with better contig binning performance, right? So what's the reason? Let's turn to the raw Hi-C contact matrix. In this matrix, each row and column represent contigs from two different species, while the genome coverage of the first species is much higher than the second one. Here, the genome coverage measures the number of copies of genomes existing in the community. We can notice how the Hi-C contacts for the first species are more abundant due to higher genome coverage of the first species. However, this imbalance can skew our results and make the clustering tricky because the clustering algorithm we use often expect the clusters are in similar density, right? Moreover, within the dashed boxes, we can see the Hi-C contacts linking contigs from different species. These are what we call the spurious interspecies Hi-C contacts and are likely from the experimental voices, such as DNA from different cells being mistakenly cross-linked. These false links can deteriorate our clustering.
I was curious, is the same difference in the overall intensity of the context seen in the shotgun sequencing data? Or is this like a distinct, are all the species in the mix in the same concentrations? Like, does this reflect the abundance or not?
This shows abundance of the species
Okay.
in the community.
I see. Okay. So it's correlated with what you get from shotgun.
Yeah.
Okay.
So we can also find the spurious Hi-C context here, right? Those kind of spurious contexts can deteriorate our context binding performance, and even worse, might lead to the incorrectly identified viral host interactions. Therefore, for normalization, we have two scientific questions. It is necessary to eliminate the systematic biases from raw Hi-C context and remove the spurious Hi-C context, right? So our Recom 2021 paper is the first one to explore the impact of systematic biases on the meta-Hi-C data analysis. And in our paper, we put forward three potential factors of systematic biases. Besides the context coverage we mentioned before, there are another two bias factors, including the context length and the number of restriction sites on context. Intuitively, longer contexts have more room for the ligation or joining, increasing the probability to create chemical junctions, while a higher number of restriction sites on context increase the chance of cross-linking, leading to more Hi-C contacts. To understand the impact of these factors quantitatively, we calculated the Pearson correlation coefficients between the raw Hi-C contacts and the product of three bias factors. The coefficients were 0.429, 0.4, and 0.184 respectively. These results demonstrated the existence of strong systematic biases for the raw meta-Hi-C contact maps. So before I introduce my normalization method, let's briefly review the linear regression, which forms the foundation of my approach. In the linear regression, we have an outcome variable y here, right? I want to predict this variable using the predictor variables x1 to xp. We also have an error term epsilon here. A well-fitting model means that the predictor variables and the error term are not related, which also implies that the residuals, which are defined as the differences between the predicted and observed values, are independent with the predictor variables. With this framework in mind, we want to construct a regression model where our raw Hi-C contacts are our y, and three bias factors are our x. After running the regression model, the residuals will serve as our normalized Hi-C contacts. The main idea here is that we want to use the residuals independence from the predictor variables to effectively remove the effects of three bias factors from our Hi-C contact data. More specifically, we use the raw interspecies Hi-C contacts, which are defined as the number of Hi-C read pairs mapped to context from the same species as our response variable. To generate the raw interspecies Hi-C contacts, it's essential to know the species of at least a subset of contexts. However, in practice, the species identity of a sample context is often unknown in real datasets. To address this challenge, we employ the text design to label some contexts at the species level by running the blast against its curated liquid hydrogen database. Moreover, we cannot directly use the linear regression in this case because our response variable is non-negative count data with excess zeros. So instead, my normalization method, named Hi-C-Zin, models the raw interspecies Hi-C context by a two-component distribution, where the counts can either be zero with a certain probability or follow a more variable pattern described by the negative binomial distribution. The two distribution variables are linked to three factors of biases using the log and logic linking functions, while the betas and gammas in the linking equations serve as the regression coefficients. Based on our statistical framework, we can write out the likelihood function, and we can use the maximum likelihood estimation to solve the regression. Finally, the residuals of the regression are regarded as the normalized by the high C context, which are free from the 3 bias factors. So this table shows the effectiveness of the normalization. The first column shows different normalization methods, while each number in the table represents the Pearson correlation coefficient between the normalized high C context and product of 1 bias factors. A smaller coefficient means that this bias factor has less influence on the high C context. As you can see, our high CZ method is the only method that can effectively eliminate all the biases we are concerned with compared to other normalization methods. As I highlighted earlier, low high C content maps do not have a very good performance in contig binning, likely due to the existence of biases. Our suspicions can be confirmed here. When we apply high CZ normalization to high C context, the clustering performance improves dramatically. This tells us normalization is key in refining contig binning and supports the idea that the bias indeed deteriorate our contig clustering performance, right? Another big issue is to remove the spurious interspecies high C context. This challenge was first put forward by David Wellman's lab from Stanford and remained unsolved for a while. He will give a simple but effective solution. This solution is based on the observation that after high CZ normalization, the distribution of normalized spurious high C context deviated considerably to the left from distribution of normalized rally context. This observation suggests that we can set a cutoff, like dashed line here, and discard all the normalized high C context below it. By doing this, we can get rid of most of the spurious context without discarding many true interactions. Although this method is straightforward, it's actually the first attempt to tackle the issue of spurious high C context. The tricky part is how to determine this cutoff, which might be different across different datasets. There is a heuristic strategy to choose the cutoff, where we first bin the contigs based on all normalized high C context, and then determine a cutoff that can remove most of the inter-cluster high C context while remaining most of the intra-cluster high C context. For example, in this case, we can choose 0.6 as our cutoff, right? Since our high CZ method can...
So can't you just, if you have the identities of your clusters, can't you just zero out everything in the inter-cluster space? Like, why do you need to remove the intra-cluster reads also? Maybe I'm missing something.
Yeah, because we don't know whether the inter-class high C context is spurious or not. So the spurious context does not mean that it's inter-class high C context. For example, if we find the interaction between the viral genome and the host genome, they are inter-class high C context, but they are true linkage, right?
There's a different application on this,
Yeah,
but in the
yeah.
synthetic yeast mixture, you know that anything intercluster is fake.
Yeah.
So you should just actually just remove anything between clusters.
Yeah.
So you're saying this is a solution for a different problem?
Actually, the spurious high C context mainly influence the detection between the host and multi elements. So those kinds of inter-cluster high C context may mean the interactions between the virus and phage.
But not in this 13 mixture that you have.
Oh, this is different.
In the 13
function.
mixture of yeast, right? Like
Yeah.
anything inter is just spurious.
Yeah. But
So this is kind of like validating an approach on some other data that's like maybe not even quite here.
This
so.
figure is not for the 13 species. This figure comes from a real data set.
Okay.
It's not the results on the yeast data set.
Okay. Because if you weren't looking for if you had no reason, if your prior was that anything intercluster is wrong, then you should be able to use, you shouldn't just have to set a threshold that's agnostic of the cluster identity, right? Like you should be able to use the fact that all of these things are in the same cluster, all these things are in another cluster, and you can just aggregate all of the reads. Like, I guess I don't understand why you're doing a per contig pair threshold instead of a per cluster pair threshold.
Yeah.
Does that make sense?
Yeah, I think so.
Okay. Is there a reason why a per contig pair threshold would be better than a per cluster pair threshold? Like aggregate all of the reads in that whole inter-cluster space, right? You have one number. Shouldn't that separate like entirely from the distribution of intra-cluster reads?
So you mean you just calculate the average connection from each contig to the other
From each cluster, yeah.
each cluster, right?
Yes.
I'm not sure about this question. Actually, this strategy is put forward by another paper, not what I put forward.
We can talk more about it later.
So see, all things how hydrazine can help to remove spurious hydrazine linkages. It has been used to construct interaction networks between hosts and mobile genetic elements. For example, the hydrazine has been applied to generate the viral host interaction network for microbial community from the dense hydrothermal mass and construct the plasmid host interaction network from the human gut environment. Although our hydrazine model has been widely used in meta-Hi-C downstream analysis, we have to admit that hydrazine is not a scalable normalization method. There are two problems contributing to the scalability challenges faced by hydrazine. The first problem is that because hydrazine works with the Hi-C context between contigs, the complexity is O(n) squared, where n is the number of contigs. This indicates that hydrazine will spend a lot of time on large meta-Hi-C datasets. Moreover, let's look at the right side of the regression formula in hydrazine. We have three predictor variables: the number of restriction sites on contigs, contig length, and contig coverage. The first two are straightforward and can be directly obtained from the contigs DNA sequences. But contig coverage is tricky. It measures how many copies of contigs existing in the community and are always estimated by mapping the shotgun reads back to contigs. However, this estimation process is very time-consuming and memory-hungry. So these challenges encourage us to develop a more scalable but effective normalization method, right? And in our Nature Communication paper published last year, we give our solutions to these two problems. The first issue is that the complexity is O(n) squared. To tackle this, we shift our focus from modeling the individual intra-species Hi-C context to modeling the sum of all Hi-C contexts for a given contig with the rest of contigs in the sample. Here's the trick. We take the log sum of the raw Hi-C content matrix, excluding the diagonal entries, and assume this sum follows a lecture binomial distribution. This sum, which we call the MI for each contig I, becomes our new target for modeling. Then we bring in a new regression formula and links the mean of MI, denoted as mu I, to the three factors. This change simplifies the model significantly. Instead of computing a relationship for each pair of contigs, we are now looking at the relationship for each contig individually. As a result, the complexity drops from the O(n) squared to O(n), which is a huge improvement in terms of scalability, right? The second problem is that it is time-consuming and memory-hungry to estimate the contig coverages. So my solution to this problem is to represent unknown contig coverage using observable variables. Specifically, I modeled Ni, the diagonal entries of the raw Hi-C content matrix H, as our response variable in a negative binomial regression, where the three bias factors serve as predictor variables. So remember what I say about linear regression? If the independent or outcome variable y can be fully or perfectly explained by the dependent variables, the conditional expectation of the residuals equal to a constant, right? We apply the similar logic here. If you assume the Ni here can be fully or perfectly explained by 3 factors of biases, and under some certain technical conditions, we can also assume the residuals of this negative binomial regression as a constant. And through some simple mathematical transformation, we can express the coverage for each contig I using another 2 observable variables, Si and Li, as well as the Ni, which can be directly obtained from the raw Hi-C contact matrix. So based on our previous solutions, we have refined our approach into a new method named LOM-CC. LOM-CC plugs the approximate expression of contig coverages in the second solution into the regression formula in the first solution to get a final regression model. In our final model, we are working with μi, keeping our complexity linear with respect to the number of contigs. On the right side of the equation, all predictor variables can be directly observable. We still use the maximum likelihood estimation to solve this regression. And then we normalize the Hi-C context by dividing the raw Hi-C context by the square root of μi times μj, then applying a scaling constant to adjust the size. Finally, we prove the effectiveness of the LOM-CC normalization method on the synthetic yeast dataset we introduced before. The left table shows very low Pearson correlation coefficients between the LOM-CC normalized Hi-C context and 3 factors of biases, demonstrating the effectiveness of the LOM-CC normalization to eliminate the biases. The right bar chart shows the latent graph cluster results based on the raw Hi-C normalized and LOM-CC normalized Hi-C context on this synthetic dataset. The results are very clear. We can find that the LOM-CC outperforms Hi-C in contig clustering. LOM-CC stands out not just for its effectiveness, but also for its efficiency. The bottom table highlights the time it takes for LOM-CC versus Hi-C to normalize different datasets. LOM-CC speed is remarkable, especially on a wastewater dataset, where Hi-C took over 4 days. In contrast, LOM-CC finished normalization on this dataset in just 2 minutes, over 4,000 times faster than Hi-C. In conclusion, the LOM-CC provides a significant advancement in Hi-C data analysis, allowing researchers to normalize their Hi-C data both effectively and efficiently.
What's the meaning of the 6.5-ish in the bracket? What's the meaning of this?
Oh, okay. This is a very good question. So this means because our Hi-C so here, you can find our Hi-C requires the annotation of some contigs, right? So actually the number in the bracket means how much time you spend on the annotation with text line.
I also had a question. What are you saying? Why are you saying that it's time and memory hungry to calculate the coverage? What method are you using to do that?
You mean
in the previous slide? You say, yeah, here problem 2: time consuming and memory hungry to estimate.
Yeah. So this process is how we estimate the coverage. So we use the BWA to map the short boundaries to assemble contigs, and use the MetaBat 2 to compute the coverage using the alignment results.
I guess I'm confused as to why that's memory hungry, because you should be able to do this just like out of core. You should be able to estimate coverage just by going through the chunked table of contact counts, like an IJK table, and just.
No. Here the coverage here, we use the short boundaries instead of Hi-C reads to calculate the coverage, because Hi-C reads are biased, right? They're only generated from the regions that have the restriction sites. We want to estimate the coverage alongside the contigs. So you use the shotgun reads instead of high series.
So you're saying you're trying to use kind of the abundance
Yeah,
of the
yeah,
of
yeah.
the contig
Yeah, yeah,
as a
yeah. They
normalization.
have the coverage of
from
just
the-
abundance of contigs.
from the-
It's not a high-C coverage that
Yes,
you
yeah,
understand
yeah,
in
yeah,
the
yeah.
individual high-C genome.
But did you try using the high C coverage, even though it's like you could still use
Yeah,
it as
it's a very good question. We find that the high-C coverage is not very accurate.
okay, and it's not useful as a
Yeah, yeah.
regressor. Okay, interesting.
So given that lone CC significantly improves contig clustering, a natural progression is to develop a contig binning method that can average the strengths of lone CC normalized high-C context. However, if we only rely on high-C context to do the contig binning, as done by current binning methods, some very important biological information embedded within contigs will be ignored. In fact, high-C context can be a very good metric to tell us that two contigs may come from the same genome. But are there any sources of information that indicate that two contigs do not come from the same genome? The answer is yes, and the single copy marker genes come into play. In short, the single copy marker genes are unique genes and only exist once in a species genome. Therefore, if we find the same single copy marker genes from different contigs, it suggests that these contigs come from different genomes. For example, we can find that both contig 1 and contig 2 have the single copy marker genes SMG2, right? This indicates that these two contigs come from different genomes. So one straightforward idea is that can we combine these two powerful sources of information to refine the contig binning process? This inspiration led me to develop IMPUCC, an integrative contig binning approach. The main idea of IMPUCC is to leverage the combined power of lone CC normalized high-C interactions and the single copy marker genes. Initially, it clusters contigs containing marker genes into preliminary bins. Then, the IMPUCC will utilize insights from these preliminary bins to refine the contig binning process for all sample contigs. The IMPUCC pipeline contains three major steps: imputation, reclustering, and integrative binning. I will briefly introduce the main idea of each step. The IMPUCC pipeline begins with an imputation step. The goal of imputation is to amplify the cross-contig high-C signals for marker gene containing contigs. To realize the imputation, we design a constrained random walk with restart algorithm, which will limit that all the random walks can only start from and return to the contigs with marker genes. In this way, we can predict the missing interactions between the marker gene containing contigs and other contigs. We can see that after imputation, the cross-contig interactions among marker gene containing contigs become substantially stronger and more pronounced, right? In the second step of our IMPUCC pipeline, we utilize the enriched high-C context from the imputation step alongside the single copy marker genes to pre-cluster marker gene containing contigs. Our aim is to generate preliminary bins as accurate as possible. There are two guiding principles in this process. Firstly, if two contigs share the same single copy marker genes, they must come from different genomes, right? This principle is ensured by the property of single copy marker genes. Secondly, if two contigs do not share any marker genes, but are linked by very strong high-C signals, then these two contigs may come from the same genome. Based on these two principles, we can generate accurate preliminary bins. The final step is the integrative binning. So we have got our preliminary bins from the previous step, and we are confident that the contigs from the same preliminary bin should come from the same genome. Then we can merge contigs from the same preliminary bins into superloads in our high-C contact network. For example, let's say if we have two preliminary bins, we will then merge contigs 1, 4, 7 from the preliminary bin 1 into superload PP1, and do the same thing for the preliminary bin 2 and get PP2. In this way, we can get a new network where each load or each superload represents a collection of contigs. On this new network, we can run the latent graph clustering algorithm to group all the sample contigs into draft genomic beans. We bench MQCC on both MORC and real meta-HiC datasets. The MORC meta-HiC datasets are created by combining the simulated HiC reads with real shotgun sequencing data from a manually created microbial community with 65 long species. The shotgun data are obtained from the Illumina short reads, Lalaport long read, and PacBio long read platforms. These datasets named MORC Illumina, MORC Lalaport, and MORC PacBio, which consists of a combination of simulated HiC reads and real shotgun reads corresponding to a specific sequencing platform. We also benchmark MQCC on a real SheepGuard meta-HiC dataset, where the shotgun data are obtained from PacBio Hi-Fi long read platform. For MORC datasets, we directly evaluate the quality of each draft genomic bean by comparing the contig length to long reference genomes, with high-quality genomes requiring higher than 90% completeness and lower than 5% contamination. For real datasets, because we don't have the ground truth and the reference genomes, we use the check-M to estimate the completeness and contamination of each bean and define the high-quality genomes using the same criteria. The figure on the left highlights the superior performance of MQCC across 3 different MORC datasets. Especially in the MORC PacBio datasets, MQCC retrieves over double the number of high-quality genomes in the second best method. This trend holds true in practical application, as demonstrated by our analysis on the SheepGuard meta-HiC dataset. MQCC still significantly outperforms other methods in recovering the high-quality genomes. Impressively, MQCC constructs a total of 408 high-quality genomes from the SheepGuard meta-HiC dataset. This is a record-breaking number for reference quality draft genomic beans extracted from a single microbial sample. We finally performed the taxonomic annotation on high-quality beans retrieved from the SheepGuard dataset using the GTDB. We find that MQCC is the only method that can recover the genomes from the genus Bacteroides, known for affecting the intestinal immunity. We also find that MQCC can recover 5 genomes from the Alistifis genus and that could not be allocated at species level, suggesting the potential expansion of the species diversity. The MQCC has provided us with significant advancement in convening from meta-HiC data, but it has its limitations. Specifically, since MQCC relies on the single copy marker genes, it cannot be applied to bean the viral contigs because there are no single copy marker genes embedded within the viral contigs. Moreover, other HiC-based bean methods that only rely on the HiC networks also perform badly because the HiC content network for viral contigs is very sparse and cannot provide enough information for precise beaning. Recognizing this gap, we have created a pipeline dedicated for viral contigs that successfully retrieves complete viral genomes from metagenomic HiC data. So we call this method ViralCC. In ViralCC, we want to solve the challenge of the sparse HiC network among viral contigs. The main idea is that we can find other sources of information in viral contigs other than the direct HiC interaction. So let's look at this figure. This figure shows the viral contigs from a virus that is infecting a bacterial cell. When we do the cross-linking, you can get HiC interactions inside the phage, right? And this HiC interaction provide us with a sparse HiC interaction graph. However, you can get more than that. Apart from the HiC interaction within the phage, we can also observe the HiC interaction between the viral contigs and the host genomes. In other words, if 2 contigs are really coming from the same phage, they should interact with the same host through the HiC linkages. So based on this property, we can construct the host proximity graph, where those are viral contigs and the edge shows the 2 viral contigs share the same host. Then our BioCC utilizes both sources of information simultaneously by concatenating these two graphs, and does the latent graph clustering based on the integrative graph to generate the draft viral genomes. We benchmark BioCC on wastewater and high-C dataset. In this dataset, both shotgun and high-C libraries are sequenced using the Illumina short-read platform. After sampling, we use the viral sorter to detect the viral contigs and bin these viral contigs into draft viral genomes using BioCC and other binning methods. We use the check-V to evaluate the completeness of draft viral genomes and further allocate the draft viral genomes at a family level by comparing the genes on viral contigs against the curated viral protein database. The left figure shows the completeness results evaluated by check-V. We can find that BioCC recovered much more complete viral bins compared to other methods, especially for the deep blue bar, which represents the draft viral bins with very high completeness. We further allocate the draft viral genomes at a family level, and the right figure shows the distribution of viral bins across different families in the wastewater dataset. And we can observe a comparable abundance of double-stranded DNA viruses, malviral and cycloviral. In contrast, the gut environments, such as human gut, are typically only dominated by the cycloviral. In conclusion, let's go back to the three questions I put forward at the beginning of the presentation. For the first question, who is in there? InPuCC and ViralCC can help solve this question. InPuCC combines the power of high-C context alongside single copy marker genes to bin contigs into draft genomic bins. And ViralCC is dedicated for viral contigs and can recover the draft viral genomes. For the second question, what are they doing? Our LOM-CC and high-C contribute to this question. These two methods can help us remove the spurious high-C interactions, and the rest of the true interactions can be used to reveal the true linkage between the mobile genetic elements and the hosts, or reveal the micro interactions. However, we still have another question. How are they doing? This is what I want to contribute to in the future. To identify the disease-associated viral host interactions, a large-scale database of viral host interactions from the healthy human gut is needed as control to conduct the correlation analysis. However, we don't have such kind of database currently. So in M1, I plan to construct a comprehensive and high-quality phage host interaction database using public healthy human fecal and high-C samples. However, it's impossible for the database to be completely incomprehensive. For example, when a new phage that is not included in the database comes in, how can we know which host this phage can infect? This motivated me to develop an advanced machine learning model that can infer the phage host collections in healthy human gut for virons that do not currently exist. And this machine learning model can be trained using the database we construct in M1. Finally, I want to create an accessible and convenient platform where users can submit their metagenomic sequences to obtain and visualize the phage host insights. Okay, for my acknowledgement, first of all, I would like to extend my sincere gratitude to my advisor, Dr. Sang, for guiding and supporting me during my Ph.D. journey. I think his passion and dedication in research has deeply affected and encouraged me to explore further in academia. As I progress in my career, I think Dr. Sang's example will always guide me. I would also like to express my sincere gratitude to Dr. Waldman and Dr. Foreman for their support and guidance when I pursue my academia career. I would also like to thank Dr. Foreman for his valuable comments on my research projects. I also want to thank my dissertation and qualifying committee members, Dr. Chen, Dr. Hildenberg, and Dr. Pano. And I want to thank Dr. Ross for his suggestions and comments on my job talk and interview. And I want to thank my former and current lab mates and my old friend in China and relative new friends at USC. It's exactly you that makes my PhD time both enjoyable and memorable. Finally, I owe my deepest gratitude to my dear parents, my mother, Liu Junfeng, and my father, Du Lizhi. From my childhood, they have been my role models, teaching me how to become a good person. And they have consistently encouraged me to follow my dreams and passions. Without their encouragement, I could not be here. Thanks for coming here, and I'm happy to answer any questions.
Okay, so I think we can get some questions from the student first, and then we release them, and then the committee will ask questions.
Why did you choose that CINB model?
Yeah, this is a very good question because sorry, it's not shown here. Why? Okay. Yeah, because it depends on the distribution of the response variable, right? Because our response variable is non-negative count data with exact zeros. So depending on this distribution, we use the ZNB model.
So I remember you have a slide about retrieving the genomes from the bacteroides from the sheep gut, right?
Yeah.
So your tool is the only tool that can like retrieve that genome.
Yeah, actually, so other mini methods can retrieve this genome as well, but they are not in high quality.
Okay. So this retrieval uses the information both from shotgun and metagenomic, right?
Yeah.
Do you compare like other tools that only use the shotgun data? Because I remember there are a lot of such tools.
Yeah, that's a very good question. So actually, you can see this benchmarking here. The VNB is the only method that uses shotgun sequencing, or only use shotgun sequencing, and other methods use high-C information.
So you can like retrieve this kind of genome more completely is it due to the high volume of that genus in the sheep gut? Because I suppose that genus is a predominant groups in the gut, right?
Yeah. Actually, as I mentioned, other methods can also retrieve this genus because it's predominant. But all our methods can retrieve high quality genomes. We have a very strict criteria for the quality of the genomes we retrieve.
Thank you.
Okay, thanks for the questions, and you are. Right. Now we can open questions for the committees.
There are some people online. Okay. Here we are.
Yeah, I think probably you can stop the recording.
Oh, yeah. Sorry. I can start recording here.
Looks like it's just